Okay, class, where we are now is to talk about the parenchymal cell of the nervous system, which is the neuron. Okay, special characteristics. It may have to go 100 years or more. Since they don't do mitosis, if you live to 100 or over, that's your neuron you got to stay. It's amitotic. After a certain age, early age, they don't multiply. They just get a little longer. High metabolic rate. Remember, they do action potentials. A lot of ATP. Needs a lot of ATP, in the Krebs cycle, using glucose. The cell membrane, plasma membrane, functions in electrical signaling. We talked heavily about that in electrophysiology. Cell-to-cell interactions during development. Okay. So here's the parts of it. Okay. You have a cell body, which is also called the soma for body, or the perikaryon. Now remember, karyon was an old name for the cell nucleus. So an area around the nucleus. It's the biosynthetic area of the cell. This is where you make all your neurotransmitter substances we talked about and everything. And then it transports it. So it does all the making of stuff up here. Spherical nucleus with a nucleolus. Remember, the job of the nucleolus was ribosome production. Well-developed Golgi. Remember, Golgi is to package stuff for secretion, and these cells do use that neurotransmitter substance we talked about in electrophys. Rough ER. If you got a Golgi, you need the rough ER. Rough ER is where you make proteins. Now, the rough ER, because it goes by a person's name, remember what I call eponym. Eponym was named after a person. In the early days, that's how you gave credit, known as the Nissl bodies, or some call them Nissl substance. But they stain. They stain a dark color. So chromatophilic, Latin breakdown, philic to see color on the staining, chromatophilic substance. And that's up in here and some into the dendrites. You don't see any of that in the axon there. Okay, let's go further there. Okay, so network of neurofibrils, neurofilaments up in there. Then you have this axon hillock. The axon hillock is the first area coming off the cell body where, go back to your electrophys, where the voltage-dependent gates are close enough to develop action potentials that propagate. The axon hillock is the first area where the voltage-dependent gates are close enough to develop action potentials that propagate. Back here is graded channels. Here is voltage channels. Okay. Cone-shaped area from what act clusters of cell bodies are called nuclei. Okay, here's the situation we'll come to, but when you have a cluster of cell bodies in the central nervous system, that's called a nucleus. A cluster of cell bodies in the central nervous system is called a nucleus. A cluster in the peripheral nervous system is called a ganglia. And we'll do that a little later again. Okay? All right. So we come back. Processes coming off. Dendrites. You can have a bunch of dendrites coming off, particularly when we get into the multipolar talk. Bunch of dendrites, and they can even sub-branch. But you can never have more than one axon coming off the cell body. A bunch of dendrites, but never ever more than one axon coming off. Okay? Now, just like you had a cluster of cell bodies, Okay, class, we're back now. When you have, I'm going to come back here now. And what we had was, let's come here, a cluster of cell bodies are called a nuclei in the CNS, ganglion PNS. So I made a little chart real quick. In the CNS, central nervous system, a cluster of cell bodies is called a nucleus. Plural is nuclei. In the PNS, a cluster is called a ganglion, plural ganglia. Okay, so we go a little further. Processes. Processes would be the axon and dendrites, which I'll call fibers. Axon and dendrite, which I'll call fibers. Okay. See, let's go back. Here are the dendrites. Here's the axon. Now, to say it again, dendrites receive the signal. It brings the signal in. Axon carries it out to the next whatever, whether it be to another neuron, neuronal, or gland, neuroglandular, or neuromuscular processes. So I call them fibers. So when we come back, the processes, a bundle of processes, which I call fibers, which same thing in this case, are called tracts in the CNS. Tracts. And nerves in the PNS. So the technical definition of a nerve is in the peripheral nervous system and a cluster of the axons and dendrites. Okay? Dendrites, let's go back to our picture here. Dendrites, short, tapering, and diffusely branched. Receptive, they bring the signal in. Convey electric towards the cell body. They do graded potentials. They don't have the voltage gate stuff here. The axon, one axon, remember I told you, not no more than one axon can come from the cell body, arising from the axon hillock, the first area that starts having those voltage-dependent gates. Long axons, they can be very long. For example, in the sacral area spinal cord, all the way from that area down to your toe is one long axon. Now that cell is not getting that, it's a long cell, but it's not disturbing the surface to volume because it's so cylindrical. Occasionally branches. So in other words, even though you can only have one axon come off, this axon can branch into collaterals. Not shown here, but collaterals. Okay, so one coming off, but it can branch after it comes off, and that's what we call collaterals. Okay. Numerous terminal branches. These are called telodendria here. These axon terminals also can be called telodendria. Now the knob-like endings, see, we talked about that. The synaptic knob, synaptic end bulb, we did that in the electrophysiology section you did right before muscle. Secretory region, release of neurotransmitter, did all of that. Axon function, conducting region of the cell. That's where the action potentials can occur. And what it's doing is conducting the signal away from the cell body to wherever it's going to synapse with. Okay? All right. Molecules are moved along the axons. So in other words, the biosynthesis occurs here. Let's just put it this way. You have intermediate filaments and neurofilaments that carry stuff down. So you make your neurotransmitter carry it down along with other things. It's not just neurotransmitter. Then stuff you're bringing back, wasteful stuff, or broken down neurotransmitter comes back. So antigrade towards the axon terminal, retrograde back to the cell body for making some biosynthesis and getting rid of some waste. Okay? Unfortunately, though, coming back, riding the pipeline may be something you don't want, and that's viruses and bacterial toxins. Unfortunately. That's how they get into with certain central nervous system infections and so forth. Let's go a little further. The myelin sheath. Remember, in the central nervous system, it was placed by the oligodendrocyte, in the peripheral by the Schwann cell, is a protein lipid sheath that surrounds this, but it's in plugs. It's in plugs. And what it does is it speeds up nerve conduction. It functions to protect the insulated axon, but the main thing is speeds up nerve conduction. Okay, that's the myelin, myelinated neurons. Not all are myelinated. Some are not fast. Okay, the way this occurs, and I'm going to go to this, is this. Now I want you to think about this. Sad, and this is not a good example, but I'm going to use it. Let's say that you had snakes that were coming towards you and wrapped around your leg. Let's say one snake wrapped around your leg, and it wrapped around so tightly, almost like a towel wrapping around your leg, so tightly that it squeezed the skin, that literally the snake skin now is your skin at that area. The snake skin is your skin at that area. That's literally how tight this is. So the Schwann cell approaches the axon. The Schwann cell approaches the axon. This is one Schwann cell approaching the axon, wraps itself around like a towel or a snake around there. So much so that now the cell membrane, listen to me, is not any longer underneath here. If I said draw the cell membrane now, it would be here and then the outer of this. So it's the outer now of this becomes the cell membrane of this axon. Then several wrap on, but they leave a space in between, which is going to be important. So that's how the myelination occurs. That's how it occurs. And it wraps real tightly. Now, this cell has a lot of sphingomyelin. Go back to your biochemistry, my chapter 2. Sphingomyelin is a lipid. So this cell has a lot of that. So when it wraps around, it puts a lipid coating covering. Now, when it wraps around, it closes up those voltage-dependent gates. Remember, the gates are water-soluble holes. You wrap around and plug the gates up, this towel, in that area. That's going to be important as how when we get ready to explain how this does its job. Then you wrap, another one wraps or wraps. But the important thing is it's not, you don't take one long wrap, it's in pieces. And in between the pieces is something called the internodal regions, or going towards the eponym, the nodes of Ranvier. Internodal, internodal, internodal are also called the nodes of Ranvier. Okay, I want you to read on it now. Protein lipoid, it functions to protect. So we go here, wraps many times around the axon, myelin sheath, concentric layers of Schwann cell membrane. The neurolema now, which is a fancy name for the cell membrane, the neurolema, fancy name, is actually in those areas the outside of this. So neurolema, but now it jumps and becomes that. Okay, it wraps so tightly. Nodes of Ranvier. What did we say about that? The nodes of Ranvier, and this is important, that one snake grabs another one, Schwann cell in this case, but you leave a space in between called the internodal regions or the nodes of Ranvier. Okay? And we'll talk about that. Okay, as to how it works. Okay. So some neurons are unmyelinated. If they are, then what happens is the conduction is not as fast. One Schwann cell may end up grabbing several at the same time. It may grab several at the same time. Okay. All right. So we go a little further. White matter and gray matter. That's my next subject. So we'll end right here.